On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
In this episode of Life Science Success, my guest is Kris Franklin, and I'm looking forward to the chat with him. But before we hop into the interview, let me just play a quick message from our sponsor.
Being at the epicenter of the next breakthrough in biotechnology. That moment could happen for you this October at Bayou on the Bayou 2025, where Southern innovation meets scientific excellence in the heart of New Orleans. Over 300 of the brightest minds in biotech are coming together for 2 extraordinary days in New Orleans. We're talking groundbreaking research from powerhouses like Tulane, Auburn, the University of Mississippi, Xavier University of Louisiana, and LSU Health. Expect revolutionary technologies, cutting-edge research, and industry-shaping expert panels that you won't find at any other event, plus networking opportunities that could transform your career. This isn't just another conference, it's an intensive 2-day summit where real partnerships form. We leverage the cutting-edge Partnering One platform to ensure that you'll connect with exactly the right people, whether you're seeking investors, research partners, or your next big opportunity. The future of biotechnology is being shaped right here in the broader Gulf South and having a global impact. Will you be part of it? Mark your calendar for October 28th and 29th. Head to bioonthebayou.com right now and secure your spot for the most exciting biotechnology event of 2025. The next breakthrough could be yours. Bayou on the Bayou 2025. Register today.
In this episode of Life Science Success, my guest is Kris Franklin. He's a graduate from Vanderbilt with nearly 20 years of experience in pharmaceutical, agricultural, and clinical diagnostics. Mr. Franklin founded Core Consulting in 2014, and he helped Clinicore Labs deploy LDTs in the field of toxicology and pharmacogenetics. With that, let me just welcome my guest here. Hey, Kris.
Hey, Don, thanks for having me on.
Yeah, it's great to have you on, and I look forward to our interview. Obviously, I've done quite a bit in diagnostics myself, and so I'm looking forward to sort of diving deep with you in terms of your background, but also the one thing that I left off of that quick introduction is that you also founded Clinicore Labs. You were telling me before we got started in 2017, right?
Yes, sir. So that originally, I came up with the very first idea of doing some type of small volume blood architecture to measure tests, because back then, I remember looking at it and thinking, well, LabCorp and Quest kind of eat up the bulk of diagnostic testing. They've got their little draw sites everywhere, and there's no way for a small lab to come in and try to compete with them because they've got it on the corner everywhere. So I was like, what if I could just figure out a way to collect it from a fingertip, make it simple? Even if I couldn't run all the tests, it would allow me to not compete head to head with the 800-pound gorilla, but be able to offer a technology that was different. And so that kind of started in the consulting. And eventually I'd had my first enough data and validation behind it to have a provisional patent in 2017. I put that patent together with a business plan, and investors saw it and basically, in a sense, Shark Tanked it and sold 25% of the company to a partner for $3 million. So it gave us an initial seed valuation of $12 million. And in probably September of 2017, we first began building out the space. And eventually in February of 2018, we were live and had validated ourselves for clinical samples.
That timeline really helps overall. As we sort of get started here, it would help as well to just maybe explain what is it that brought you into life sciences? What made you, be interested in this field? And then ultimately what led overall to your being in diagnostics of all places?
Right. It was I'm one of those, I'm probably a jack of all trades and a master of none. I've just enjoyed, I started out, I was big into working out, big into fitness, and I got certified as a personal trainer. This is right out of high school. I love the gym, and I eventually decided to go to school to become a physical therapist. And as I started getting into the introductory biology, some of the cell biology courses, anatomy, physiology, what I found was like some of the things that were what I would call bro science or gym myths didn't really hold up to what you would actually find in science. Like biochemistry wise, it didn't make sense. And so I got more and more curious about the science because I was always in this urge to try to integrate the two together. Part of it for just self-serving purposes to how far could I push my body? How far could I improve health as a whole? And the more I understood, the more I felt I would do. So I ended up moving from PT to pre-med, and I ended up in all degree disciplines as chemistry. And to be honest, I wasn't very good at it. I got my teeth kicked in a lot in chemistry, but this is probably mid to late 90s. And if you remember during that time, we had the World Human Genome Project, and they were in the process of trying to sequence the first human genome. And a guy by the name of Craig Venter, who was part of the project, came up with a different way of trying to sequence. And he went to the World Human Genome. He's like, hey, guys, we could do it this way, and it'd be a lot faster. And the whole world was like, no, man, you don't know what you're talking about, this and that. And so, in a sense, while he's riding in the car with him, he almost jumped out ahead using his ragtag scientist because he was able to see the problem from a different perspective. And I watched this one guy beat the entire world at sequencing the human genome. And I was like, man, if you could just learn the disciplines of science, it'll allow you to push the boundaries and actually explore. So it was almost my ADD brain thinking, man, if I could just buckle down and get a grip on the foundational rules in science and things that are in play, then I can figure out how to push the envelope wherever you go, because it gave you a chance to make this kind of creative expression that really other disciplines didn't allow you to go. And so that just became kind of an epiphany and awe-inspiring to me. And so that's kind of what's really got me in the track of, instead of going pre-med, I decided, hey, look, I'm going to stick with just science and just see how far it could go. And the way I looked at it was, in Louisiana, being here from the Bayou, there's chemical plants everywhere. And I was like, well, if I don't get into med school or whatever, then a chemistry degree is guaranteed to find me a job somewhere when I get out of school. So that became kind of the choice. And I guess a certain point about my senior year, I was trying to graduate on time. And, you got P-Chem or quantum, you've got biochem, and I've got all these upper level chemistries. I was doing undergraduate research as well as tutoring for organic and general chemistry. And I don't know, I joke about it. I don't know if it was an aneurysm but it felt like my brain popped at a certain point. And at that point, it was almost like chemistry and science became intuitive. I'll use the example from like martial arts. If you go take 2 weeks of karate classes and you get in a fight, chances are you're going to get your brains beat out because everything you're doing, you're having to think, okay, block, do this. But if you've got somebody that's been doing martial arts for decades, then it becomes internalized and it almost becomes intuition. And at a certain point in my senior year, it felt like my brain almost became intuitive about science, where I could see something and almost react with it with a very good hypothesis to test and work my way through. And I just like the ability that science always offered a problem to solve. And by my personality and by nature, I always look for problems to solve. And considering science is nothing but problems, it was kind of like, hey, here's an entire farm of problems you can just work through and kind of test your ability. And so that became kind of the passion, was like, hey, let's find a bigger challenge. Can I find a bigger problem to try to solve? And that's kind of where it kind of began. And I think at the end of grad school, I'd gotten excited. Pat did well enough on the MCAT that I got accepted to LSU Shreveport to go to med school. I was accepted to Texas A&M for graduate school in chemistry, and as well as Vanderbilt for chemistry. At Texas A&M was actually ranked higher in chemistry, but the fields of study that they were predominantly in were material science and physical chemistry. And I'll be honest, I hated physical chemistry. Like that would have been a class, every chemist that's been through quantum or statistical thermo, I always like to say, if you could do it without the math, it would have been like sitting down watching an episode of The Twilight Zone, because you're hearing it, it's tickling your brain, but then they're like, okay, now we're going to do the math to prove it. And 4 hours later, through path integrals and differential equations, you've suddenly found out why the electron is sitting in some field. instead of being a particle unless you look at it, and all this type stuff. And I didn't like the math part of it, but I loved the challenge of it. And so I ended up going to Vanderbilt, where they were really had this kind of collaborative approach to everything. They looked at it like whether you were the chemistry department, physics, everybody was trying to tie into the medical research. So if you were doing chemistry, they were trying to find a partnership within the medical school to do some type of medical research. And to me, that was absolutely amazing. And I remember in my PhD qualifying exam, my PI directly, we were talking about it, and he said, "Well, Kris," he said, "the really big no single discipline can solve the really big problems." He was like, "It is always a collaborative approach." He said, "Because the big problems require big answers, and no single discipline has the capability to deliver that." He said, "So, and we were doing cancer research at Vanderbilt, and we were looking at site-specific, stereo-specific adduction of lipid peroxidation products on the minor groove of DNA, which, why was there a repair mechanism? Was there something structural?" So we had everything from structural biologists using high field NMR to get solution structures of these oligomers. We were transfecting them into cells, forcing cells to replicate, and then going back and sequencing the mistakes it was making. And I was doing the synthetic work to actually put these things together. And so that was kind of the graduate part, but you got to see this really collaborative work to go, hey, let's go solve a really big problem and we'll work together. And so we had 3 PIs. We were all funded by the NIH and pretty well funded at that because it was cancer research. So it was just kind of a progression. And I think that was one of the big ones that I picked up, was that in grad school was this idea of collaborating, having, instead of being a master of one skill, if you had a concept or an understanding of multiple disciplines, you were able to accomplish more. Even if you weren't the best at one thing, if you understood how molecular biology was going to use some of their techniques, then, and you understood the chemistry, the kind of the backbone of what was going on, that information would allow you to aggregate things and come up with a better solution or a better way of doing it. And I think that really just kind of inspired me looking at cancer research. And I think what really got me was I started looking around then as a challenge. And I thought, man, what would be the brightest place? I thought of like, what is the NFL for scientists? And to me, that was the pharmaceutical industry, because you would go look. And I remember when I started at the end of my grad school, I started going on job interviews. And I remember I went to Pfizer, Bristol Myers Squibb, Merck, Valiance in California. And I remember sitting down in the interview process and I'm like, these are the brightest people on earth. Like I remember thinking that I was like, they're seeing solutions to the problem I can't, meaning that I couldn't even see the problem. And they already had the answer to the problem I couldn't even find yet. And so that to me was awe inspiring. And so I really wanted to find out, was I capable, was I a big fish in a little pond, or could I get out into deep water and really try to solve and work with what would be the brightest minds out there? It's kind of a means of challenging yourself. How do you get better unless you really take on tasks that are beyond your current skill set? Let's say you've got a current skill set of 5. Well, if you try to go after something that's 6 or 7 on a difficulty scale, you're either going to fail or you're going to adapt and develop better skill sets to try to tackle that problem. And so that was my goal, kind of been my career all along, was how can I find the next mountain to climb, if you will.
That's really a really good answer. There were a couple of things that came to mind. So as you're describing some of your chemistry experience, I remember in quantum physics, essentially every answer ended in some level of decimal point, and every answer, whether or not if you had it right or wrong, was whether or not if you had the third level of the decimal point correct or not. And I mean, it was very frustrating for me as well, because it was like, well, look, I mean, an early rounding problem in my calculator could quickly, become a wrong answer then. And so,
Absolutely.
similar sort
My
of situation.
PChem professor had a knack for confusing the units. Like he would start in it and he'd be like, all right, we'll calculate the energy, but he would purposely give you some units that didn't match. So if you didn't catch it, you'd go through everything correct and solve it, but your answer would be wrong because you weren't of that dimensionally analysis of the units, and you'd recognize it when you integrated it, you did this. And oh, I know. Like it was almost, I feel like physical chemistry is almost like you go through 3 years of chemistry, you get to P Chem, and they're like, okay, this is what's really happening. it was real cool that we told you some approximations. We told you about the ideal gas law, and then you get to PChem, and they're like, yeah, there is no ideal gas. You're like, wait, what? And then, all your other chemistry is like, your electron's a particle. And then you get to PChem, and they're like, well, it's really a wave function. It's only a particle if you look at it. And you're like, wait, what? And so it's almost like science is like an onion. You're peeling off the layer, and it can't help but bring tears to your eyes, whether it's joy or sorrow. I think on the other side of it, after you're out of it, it becomes joy. But in the midst of it, you got like 10 or 12 kids suffering through quantum mechanics, and you're just like, I just need to complete this so I can graduate.
Right, right. That's the advantage I feel like a mind has whenever you start thinking about, look, one of these days I'll be in the business world, whether or not if I'm actually applying this the same way, at least I have the background for making that decision once I get out in the world. But yeah. So I guess talk a little bit about your transition as well in terms of, you have a pretty broad past as you described, but overall, you transitioned as well to clinical labs and diagnostic testing. How did that happen, and what essentially put you in this space?
All right, so it kind of like so. At first, I started in pharma. I was doing radio-labeled drug metabolism. We were making tritium, sulfur 35, and C14 labeled tracers. And part of that was you needed to understand the percent incorporation in tritium, and by doing that, you were using mass spec. And so we were coming up with different techniques. And I think one of the most valuable lessons I learned at Merck was two things. One, how to work on something when there isn't an instruction manual. Because just about everything you're doing in pharmaceutical industry, at least it felt for me the 4 or 5 years I was there, was that there is no instruction manual, and you're trying to accomplish something that nobody else has done before. And the only kicker is it's also product driven research. So they need it yesterday. So it was like, in academia, you're like, ooh, I created a new reaction. I'm going to go give a talk. I'll put a poster up and show everybody what I did. It was almost like you created that reaction just so you could complete your project on time, because you knew that the ultimate goal was right around the corner. And if I was making a tritiated tracer for an autoradiography study where they were going to map a rat brain, well, they had already had the veterinarian, the people, the anesthetists, everything set up, the animals placed. So it was up to me to figure out how to make it, purify it, prove it was pure, and then get it turned over to the next phase of the study. Because the only time you really heard about things was when you didn't deliver, because then it created a log jam, and everybody wanted to know who was the weak link in the chain. So that idea, that concept of both what I say is product driven research, is the idea is you're acquiring information, but you're acquiring information to deliver a product to market. And so that kind of creates a crunch timeline where you don't become just as inquisitive as like a natural scientist, like, oh, I want to know the whys. You're like, I need to make this work. I'll understand the why just so it'll work. And then that was something that really helped me. And then to watch people work where there isn't an instruction manual, there isn't like, open your book and make this drug. Well, who's made it before? No one. Oh, okay. Well, that's good. And you're going to put tritium on it. Where are you going to put it? Well, it can't go here or here because cytochrome P450 is going to metabolize it. So you're going to have to incorporate tritium here. When do you need it? Well, I'm starting on it on Monday. Well, when do you have to have it? By Friday. So you need to figure out how to make it, purify it, prove that it's pure, and get it shipped over so that it can go out to the study. And that type of rhythm, it kind of forces the brain to think quicker. And it was just great interacting. But I think that kind of started it. And I think the only reason I left pharma, honestly, was it was in New Jersey. And I'm not knocking New Jersey, but I'm from the Bayou, man. I'm from Louisiana. So I was 1,000 miles from family. It was cold. It was expensive. And at a certain point when my daughter was born, I wanted her to grow up around her cousins. I wanted her to be able to, my dad was trying to fly my mom up like every other week to try to see the grandbaby. My daughter was convinced that her grandparents lived at the airport because that's where we always went to go pick them up from. And so it was kind of a reboot for family. I relocated back to Louisiana, and I took a job in the agricultural industry. And it was basically everything coming in and out of the port of New Orleans has to have a third-party certificate to show that, there's either free of metals, there's no benzoate pyrene. Turns out some of the tests I developed, and what was really cool about that job is while it wasn't I would say a a great job. It was an okay job. What I found was in that job, more than synthetic chemistry, I had a real knack for developing analytical systems. And in fact, the lab, when I first got there, was about 90% of their business was just basic routine assays. What's the protein in this grain? What's this? After about a year and a half of being there, about 50% of their business had become esoteric assays that I kept developing. And so
Mm.
what would happen, people would go, hey, can you do this? And so I developed a whole suite of tests for authenticating olive oil, because turns out about 75% of the world's olive oil is counterfeited. And so I had created all these elaborate tests where I could tell whether it was extra virgin, it was refined, whether it had been blended, what oil it had been blended with. And so an entire map so that I could authenticate olive oil. And we were one of the only labs in New Orleans. So suddenly that lab's business grew on esoteric tests that I was developing for it. I also learned, based on the way the owner ran that company, that if I was ever a manager, I wanted to be nothing like him. And
Mm.
so, not it just, man, look, like the guy had been there, his business had been there for half a century, and he'd let you know from the get go, we were here before you got here. We're going to be here after you're gone. If you don't like it, you can just get out.
Wow.
And he didn't mind cutting his hand off to spite his face. It didn't matter if you brought value. If he got upset, he would do it. And so I was like, well, if I'm ever a manager, I'm not going to be that guy. And so those were the kind of lessons, but I found out I had a real knack. And then in 2012, a doctor, because this is the what I would call the gold rush, a lot of people were trying to open up toxicology labs, basically for drug testing, compliance, and not so much as like pre-employment, but anybody that was prescribed opiates had to be monitored to make sure they were really taking their medication. And reimbursements from Medicare and other payers were at that time pretty good. So there was a doctor that was about to open up a lab, and through a happenstance meeting, he offered me the position. He said, Kris, I want you to come build my lab. I didn't even know what CLIA was. Didn't know what COLA or CAP was. I knew nothing about what was clinical regulations. And but to me, again, I'm a mountain climber. I like challenges. And so I remember my wife at times, she was like, I was debating on it, and she was like, you know, you're going to take it. And I was like, what do you mean? She goes, because you always like a big challenge, you know. And the other lab had given me a good raise. They'd given me where I could finally live comfortably. And I was like, you know what? I remember thinking, I was like, if this doesn't work out, that's okay. I said, I'm young enough. I'll just reboot again. No big deal. And I remember sitting there with nothing but a folding chair and my personal laptop. And piece by piece, I built out a confirmation assay on mass spec that could do about 40 drugs in urine, then an immunoassay that could basically show that the urine wasn't diluted or adulterated, and it could screen. And within about 3 months, I got that first lab live. And I was like, wait, not only can I build analytical systems, I can build labs. And I was kind of impressed. And then within about 3 months, the doctor wanted to relocate and make it bigger. So then he had me, while I was managing that lab, building another lab in New Orleans for him. And that one was bigger. So I ended up managing one, building the second lab, got it operational within about 3 or 4 months. And that one, I started building better methods. And then he wanted to do pharmacogenomics. I built oral fluid drug testing as well. And it was a way to really test your skill. Like I didn't have the burden of the financial part or handling that. It was just like a scientist. And so for him, if I would have been like, I need 2 leprechauns and a unicorn and I can have this method built, he would have had people scouring the earth, and I would have found a leprechaun chained to one of the instruments the next day. Been like, Kris, we don't know what you need a leprechaun for, but we've got it. Get to work. And I would go build it, whatever was needed. At the same time, I enjoyed it. I liked the challenge of it. I worked myself to the bone. But one of the other things I started realizing, the lab grew to where it was generating about $25 million a month in revenue. The problem I had was that they were paying me about the same salary as they were paying some of their admins and secretaries. Granted, I wasn't as pretty as they were, but I wasn't getting paid. I was like, at that moment, I had another one of those epiphanies, and I was like, you know what? I have no problem making other people rich with me. I have every problem with making them rich without me.
Sure.
And so at about 2014, I was chief scientific officer of the lab. I resigned. And I decided, I was like, you know what? I'll starve in the street before I go make somebody else a multimillionaire while I'm broke. And I started my consulting business. Fortunately, everybody was so interested in setting up a toxicology or pharmacogenomics lab, and everybody had heard about our success. I mean, we were generating $350 million a year from a startup within about a year and a half. So my business card became, hey, don't you want to hire the guy that built the $300 million machine? And so I became Johnny Appleseed planting labs all over the country. And I would build them on whatever type of equipment you had, whatever test you wanted. I would come in, I would look at your space, I would do site reviews. This is what you need to do to get it ready. I had leveraged vendor relationships so I could come in and give you discounts on supplies, but I'd have exactly what you needed. They would order it. I'd come back in and start build their method. We'd agree. Get them all the way through their inspection. And from 2014 to 2017, start of 2017, I had built 16 labs from the ground up all over the United States. Pretty much lived in the airport, flying from lab to lab. I had two chemists that worked with me, and I was just go around building labs. Somebody wanted pharmacogenomics. I would go out there, design it, validate it, do all their validation steps, train their staff how to do it, get them through their first inspection, and then go to the next lab. And so what I'd done is I'd set up kind of recurring revenue because I could run their samples cheaper than what they could come up with. So let's say for tox, the first month you would pay me $5 per sample for the first 2,000 samples, and then it would be $2 per sample to license my method. And so I would license out. So I created mailbox money. And I think in my 2015 first full year of business, I did about 700,000 in my business and grew. But while I was doing it, I started coming up with ideas, and I'd even talked to labs. I'm like, hey, I think, as a matter of fact, I have a picture on my phone from 2014 when I resigned. A friend of mine who's a transplant surgeon in New Orleans was monitoring immunosuppressants from a finger stick. And so I went and sat with a coffee shop, and I was like, how are you doing it? And he showed me the tube, and I took a picture of it. And I remember telling some of the labs that I was consulting with, I was like, hey guys, if you want, I can change the world with this. And everybody would go, yay. And then nobody would listen to me. And I'll never forget, I'd found somebody to kind of partner with me. I built out of it. What's funny, when you talk about this small volume blood, in 2015 or 16, I had somebody partner with me. I converted a dry cleaner into a research lab, a place that was originally a space, it was a dry cleaner. I stuffed a mass spec in it because it had the power supply I needed. I got a makeshift hood, and that's what I actually built, the first prototype of the finger stick blood device that would actually measure about 12 hormones. Was the first prototype. I only could do about 12 hormones out of a finger stick, but that was the first prototype I built out of a dry cleaners. And it was funny because I'm getting invited. What's that?
I could only imagine like that rack that sort of the clothes are always on inside of the what do you call it? The dry cleaner being the spot that delivers your sample along the way. So new style automation.
Oh man, it was wild. People would come down there, and we were right down the road from we're like the same shopping center, there's a strip mall. There was a Mexican restaurant at the end, so you could always walk down there and go grab you some good Mexican food. And I built the first prototype out of that, and we kind of launched it. And then somebody tried to take it and sell it out from under me, and sold everything out from under me. And that was the start of 2017. But I had the science, and so looking at it, I went on and paid the money to get it patented, the science. And about that time, I got invited to MSACL to go give a talk. And it was one of the larger clinical conferences on mass spec. And the name of my talk was a 1 hour talk, and it was called Small Volumes or Small Problems. And the two examples, I had one where I took 65 microliters of saliva and quantitated 82 different drugs out of saliva with nanogram sensitivity. And on the other one was I'd gotten to about 14 hormones and vitamin D in a finger stick of 150 microliters of serum. And so those became the two examples. And I mean, I'm looking at it, and I've got like Russell Grant, the chief scientific officer of LabCorp, Nigel Fleming of Quest, what would be the rock stars, the legends of clinical diagnostics. And suddenly I'm walking around this room with all of these people, and I'm like, I'm about to go present. All I could think of is they're going to play Stump the Chump with me. I'm going to be about 30 minutes into my talk, and they're going to punch a hole right in the middle of one of my slides, and I'm going to have nothing left to say. And So I spent most of the first 2 days of the conference panicking, reading every publication I could find on hormones to make sure that I didn't slip up. Overall, it went really well. It was probably the more intimidating conference because there's about 500 scientists from all over the globe and all really well with what they do. But that was kind of where we got in 2017. And once we got the patent, I had an investor look at it, and he looked at the patent, looked at what we were doing technology, and about this time, if you remember, Theranos had failed,
I was just going to
and
ask you about that as well. Yeah. Yeah.
I I spent the first 2 years explaining how I wasn't Theranos,
Mhm.
and I was like, well, first off, no one gave me $1 billion because if you did, I would have built a time machine going back in time and have you give me a second billion. I said, what I built was off of just 3 million. I said, the other one was we were actually presenting it to scientists and saying, hey, look, here's our validation data. We're
Mhm.
not going to tell you how we did it, but here's the validation data. Here's where we took CAP proficiency test samples. Here's where we've done correlations with Mount Sinai Hospital, and we proved that our method works. And so we were able to prove our science without really giving it up. And it was probably the elegance in that science to do hormone quantifications on tandem mass spec. While it's considered in peer-reviewed literature to be the gold standard or the most accurate way to do it, it's also probably one of the most difficult topics or targets that you can pick. It's just loaded with problems in it. For example, like right now, because everybody has to submit the CAP proficiency testing, there for the majority of the hormones, they can't even do a round robin because there's not enough labs in the globe that can do hormones by mass spec.
Hmm.
In fact, there's only the easier hormones like testosterone, there's only 17 labs that actually participate with mass spec in the CAP proficiency testing.
Hmm.
So here it is. I'm building this out of dry cleaners, and I'm trying to go against Quest, and we're both in the same PT category. So it was unique. I guess it was really cool because I like the show Breaking Bad, and if you follow Breaking Bad, they build the meth lab in a dry cleaners, and here I am building my diagnostic test out of a dry cleaners, and
Yeah.
so
There you go.
the
So,
unique parallel to it.
so with regards to the diagnostics that you have, what role does viral transport medium play in your diagnostic process?
All right, so that one's in some of our others. So we do also do infectious disease at Clinicore, and I guess in about 2019, we were in the process of building out a PCR molecular-based assay for urinary tract infections. The idea was to be able to use the amplification to be able to determine if it was viral, bacterial, or fungal, and then more importantly, look at genes within a bacteria to determine what antibiotic resistance, if it had any, so that a doctor could make an instant decision on how to prescribe. In other words, a good example is about every UTIs, 80% of the time, doctors will prescribe Bactrim out of the gate for a urinary tract infection. Problem is, it's one of the most common to develop antibiotic resistance to.
Mm.
So being able to tell a doctor right out of the gate, hey, look, your first choice isn't going to work. You need to go with augmentins because you need a beta lactamase inhibitor along with it, just seemed like a good position. And it kind of grew. We started because urology clinics were doing a lot of hormone replacement therapy. So we were doing hormones for them. And they were like, hey, look, we've got these people that are dealing with UTIs. Can you come up with a solution for us? I was like, okay. And so we started looking around, and instead of doing the traditional culture that would take 3 to 5 days, and then you do your sensitivity and your resistance on it or susceptibility, I was like, hey, let's do it molecular. And so we started building that in 2019. And then this weird little thing called COVID happened in 2020. All of our supply chains got shut down. I couldn't get the CDC transport medium. I couldn't buy from, we were trying 10 different vendors. We couldn't even get the flock swabs that you needed for the COVID testing. So I was like, it was frustrating. And I'm at a certain point, and finally I was like, damn it, you're a chemist. You can obviously come up with something. And so I started thinking about it. I'm like, what do I need? I'm like, well, I would like to be able to lyse the cells so that if we're going to handle COVID, I don't put my staff at risk. So I wanted something that could lyse the cells, but then I also wanted something that would preserve the RNA and DNA. So I needed something that could knock out any of the RNAs or DNA polymerase chains, anything that could chop up the DNA. and then I wanted to be able to preserve the sample. And so we just kind of invented our own viral transport medium and validated it. And then we're like, well, man, we can't get a hold of these swabs. And I'm like, you know what? Viruses are really small. I bet you they're circulating in saliva. And so then we started validating a saliva based platform for being able to do COVID testing to get away from the nasal swabs. And this was in probably April of 2020, May of 2020, somewhere around there. And so by June of 2020, we were live with a saliva based PCR COVID assay that could actually even detect the different variants. And we were doing multiple targets on it so that we could get confirmation. That's how we would also know if it was a variant, because you'd see one of the proteins that we were monitoring, one of the sequences drop out, and we knew that that was one of the new variants. And so we were doing that, and because it was saliva based, a lot of the athletic departments liked it, because then you didn't need staff trying to swab people. Basically, we created little kits that would go sit at Southeastern and different universities. So whenever they needed to get so many tests a week to be able to compete in sports, what they could do is they just spit in the tube, pour the viral transport medium over, give us a call. We'd have a courier come pick it up, and they would bring it to our lab, and they would get the results the same day. So we were able, because of us, we were able to keep the Southeastern and one of the other universities' athletic programs in the fall of 2020 going. What made it special to me was I actually did my undergrad at Southeastern University. So the same place that I got my double major in biology and chemistry, and I walked across the university center, there's a picture of me posing with Roomy the lion, the mascot, because I'm there offering the technology that I created so that they could have their fall sports. So it was, to me, that was one of those like really cool moments. Now we use that viral transport medium for wounds and urinary tract infections. We don't really do any COVID testing anymore, but it works really well for all of those. So we created this kind of universal transport medium that allows us to preserve the RNA or the DNA, and it stays. I mean, we tested it up to a month where the DNA stays intact at any given temperature. So it worked out real well for us, and now we use it. So now what they can do is they'll actually swab the urine, collect the urine sample, swab it, drop the tube into our viral transport medium, send it to the lab, and within 24 hours, the doctors have got results on, okay, it's acetylbomine, and it's got resistance to trimethoprim, which means you can't going to use Bactrim. You're going to have to use probably Augmentin or Cipro or one of the other cephalosporins. And so
Very cool.
that's where our viral transport medium came in. It was more like they say, the old adage of necessity is the mother of invention. That was kind of us. Believe me, if somebody would have sold me viral transport medium back then, I'd have never invented what we had. We couldn't find it. We just couldn't buy it. Like, it was a certain point where I was so frustrated. I was almost like, screw it. I guess we're going to have to give up on COVID testing or give up on this because all the big labs had already had their lots sectioned off, and the government was kind of partitioning it to the big labs. And so the small guys were getting overlooked. We couldn't get a swab. And it was just at a certain moment, I was like, wait a minute, I'm a chemist. I can make something. And so that became the goal. And ours actually worked better because the CDC actually transported a live virus, and ours was basically making the virus an inability to replicate or spread once it was added in. So that it created that, and it made it safe for us to be able to handle around here in the lab. And so it was one of those necessity moments that we had to create it.
So in terms of Clinicore, are there any new innovative projects that you're working on that you'd like to share?
Absolutely. So we just created one. I call it One Touch. We've been alive with it for a while, and so it's basically we trimmed the volume down even further. We're able to quantify serum or plasma level concentrations of drugs with 30 microliters of blood. So it's almost a single drop of blood. It actually is collected on a dried blood spot. So we actually can do a dried blood spot. And with that, we can not only do 64 drugs, we can do 7 different hormones on 30 microliters of blood. And the bigger concept of that is what it's showing is that whether it's an exogenous target or an endogenous target, we're able to quantify that off of the fingertip and be able to get results. So let's say if you were wanting to do a pharmacokinetic study and you didn't want to be able to draw patients' blood 3 or 4 times a day. Now suddenly you could send them with those kits, prick the finger at different time points, and now the patient's able to collect from home and get it back to the doctor. one of the unique parts of that is when the local coroner's office heard about it, they were like, wait a minute, if they're doing tox off of dried blood, why don't they help us? Because the normal turnaround time on a tox study for most coroner's offices is like 8 weeks. That's why the certificate of death takes so long, is because it's all going to one lab that's supposed to be the expert. And it takes them about 8 weeks. When they found out we could do it in 48 hours, suddenly they were like, man, this is incredible. And so they moved over to us, and that allowed us to, we also validated being able to quantify drugs in vitreous fluid, which is from the eye. And so dealing with a lot of what you're seeing in these overdoses with fentanyl and things like that, we're able to quantify all those type of isomers and pieces in blood. But the other part, which I'm more excited about with the One Touch, and this is where it's kind of sprung off into the gym, is we're doing studies on athletes. And so we're looking at what if you do specific sets and reps. So now what happens, I'll have athletes prick their finger before they start the exercise routine, go through the entire routine, and at the end of it, prick their finger again. And now what we can do is we can map how the endocrine system is responding to the training. We can figure out, are you overtrained? And we found some really remarkable things, like when we look at people, athletes doing cardio. Cardio tends to maintain your cortisol levels at a very high level while you're doing it. So you did 30, 40 minutes of cardio, your cortisol won't drop. However, if you're doing weight training, we're watching cortisol and corticosteroids drop by 40% after exercise, which was really a surprise to me. I didn't expect the corticosteroids to drop, because I figured the inflammatory response of the resistant exercise was going to probably maintain cortisol levels. And we saw just the opposite. We've done it with about 20 athletes, and we saw consecutively between 30 to 42% drop in their cortisol levels with weight, whereas we didn't see a single athlete's cortisol drop because they were running. And when you think about that in terms of body composition with working out or trying to, let's say, lose fat, gain muscle, that becomes everything, because the higher cortisol is, the more likely you're going to stimulate gluconeogenesis, or the breakdown of muscle. And so the more you can drop your cortisol levels, the more likely you're going to have an actual better return on investment in what you're doing in the gym. And so what we've really done in the lab is we've partnered with both functional medicine doctors that are right on the cutting edge of doing different therapies. We're working with pro athletes. I've got pro rugby players, a few pro football players. We kind of created what I call a sports performance profile. And it really started as people just wanting to find out about themselves. And I thank COVID for that, because I think a lot of people decided to grab a hold of health and go, you know what? I'm not going to wait to get sick to go to the doctor. I want to know about myself now. And so kind of Clinicore, I created this panel that could look at inflammatory markers, all of your endocrine system. And from there, people can make decisions. And sometimes it's like, I actually started a new company called Core Health that actually works with these athletes, combining the supplements. We're doing these clinical trials. And so the idea is to say, okay, here's where you're at. Let's change your diet. Let's do this. And then we're going to monitor in 60 days. We've watched people that were almost at the point of insulin resistance, to the point of almost becoming a type 2 diabetic, shifted the supplements in their diet, and within 60 days eliminated about 70% of their insulin resistance and brought their A1C down. And so you're actually being able to give people objective measurements instead of, well, I'm feeling better. It's like, here's an objective measurement of where your body changed. And so it's kind of grown, and we're creating protocols to be able to map out the system. One of the unique things about Clinicore that made it kind of different than a lot of other labs is, so for instance, if you were to get your testosterone and you told Quest, I want you to use mass spec, they'd use mass spec. Then what would happen is they would only run testosterone. And so you'd get your results, because that's kind of the way when you're billing insurance, if a doctor ordered whatever, you can only report what the doctor's orders, because if you give the doctor more than what they ordered, it's considered an inducement. So you're very limited. However, the way our patent works and the way our hormone technology works is it's multiplex. So I'm actually mapping the entire endocrine system, every hormone from pregnenolone all the way through every minor metabolite to estriol, to the estrogen metabolites, to DHT. We're getting a snapshot, or if you think about it, an endocrinomic fingerprint of the human body. And so that's one of the unique things about our patent. Right now, I've got a database of over 100,000 patients that are stratified by age, gender, time of day, and I have their entire endocrinomic map. My ultimate goal is that database gets big, and now that with AI, is to feed it into bioinformatics and then look at ratios between hormones. And you're going to be able to find metabolic issues. You're going to be able to find new ways of diagnosing either chronic disease or current disease that people just didn't realize was available, because the data, to my knowledge, it's the only database of its kind that contains that. We're both the best and worst at what we do because we're the only that do it. And so,
So if somebody wanted to order any of those tests, is that something that's available for anybody to order, or you have to have a special way to get to them? I'm just curious.
no, so we have multiple facets. So you can go direct to consumer. That's one of the great things in recent times, I guess in the last 10 years, is the direct to consumer. And matter of fact, we do a large volume. A lot of the athletes, they're all doing we have cash prices set up so they can get it. One of the new devices that we've just incorporated, because when I originally created the finger prick, the problem was you needed about 300 microliters of whole blood. That's a lot to squeeze out of a finger. Well, we've got these new devices. I call it, basically, I can put it on your shoulder. I tap it, it'll prick the skin on the surface, and then it's got basically a vacuum, and it'll fill the tube up on your shoulder and get us one microtainer full. And with that one microtainer, I can get 35 different diagnostic tests. I can look at your insulin levels, your thyroid, all of your hormones, your vitamin D, and get a snapshot of your body. So what would happen is you could order it from Core Health. The kit comes to your house, you stick it on your shoulder, it collects a tube, you drop it in the bag, there's a FedEx label on the box, it comes back to the lab overnight with results, and then you get an electronic copy of the results within about 48 hours.
Very cool.
So we can do that for cash price. We also work with doctors. Now, if a doctor orders the test and they want to do our total endocrine or piece like that, then insurance covers it because these are known tests. And our goal was to build tests if we could find ways to improve it. And what's amazing is hormones have, like for instance, the Endocrine Society put out a note that no one under a testosterone of 300 should be measured on an immunoassay. Yet 90% of all hormone assays are ran on an immunoassay. What does that tell you? Women's range is 20 to 70, which means that no woman should have ever been measured on an immunoassay. So that means people are being misdiagnosed with PCOS, with other things all the time because immunoassays fail at that lower number. Furthermore, the traditional technology like an immunoassay, the FDA put out a warning. Turns out, good old immunoassay, when you think about a biotin streptavidin complex for making your conjugate for either a sandwich type assay, what happens is if you're taking the vitamin biotin, which is a B vitamin, it will actually interfere with the assays on immunoassay and causes false results. And what happened was people were being misdiagnosed with hypothyroidism, and it was really a biotin interference. So the FDA had to put out a warning. Now, if you look at LabCorp and Quest, they have that warning, but it's very small on their website. And unless you request them to do different, they're going to give you immunoassay. And they're like, hey, look, the warning's there. We decided instead of just putting the warning there is what if we could just beat it? And biotin doesn't really create a problem with the mass spec assay because you're looking at MRMs and you've got a very specific signal that you're looking for. So the specificity is higher. And so we've been able to do that. One of the other things we've seen in functional medicine clinic was DHEA, the unconjugated version. When patients are on hormone replacement therapy, you find their DHEA goes into the toilet because it's kind of a negative feedback. Your body goes, oh, my testosterone's high. So your body stops it at DHEA. The problem is DHEA has a lot of cognitive benefits. And we were actually the first ones to go around educating doctors to show them, like, look, if you recommend this as a supplement, 25 milligrams of DHEA for women, probably 100 milligrams for men, it's enough to balance out the adrenal glands, and suddenly the efficacy and the quality of the response on the testosterone therapy or the hormone replacement therapy is much better, and you're not having to increase the testosterone dose. That was why a lot of people, when they first got on, let's say, testosterone, they would feel great for a few weeks, and they still felt better than when they were low because of the potency of testosterone. Sure. But when their DHEA dropped off, there was kind of this overarch where they didn't quite feel quite as good as when they first got on it, and it was the body responding to it. And because everybody was using immunoassay, no one had an idea that the DHEA was being collapsed by the testosterone. And we were the first ones to come on, and I did a talk at A4M. a functional medicine conference actually showing that your pregnenolone and your DHEA get crushed when you're on TRT. And up until that point, doctors weren't even aware of it because there wasn't a lab capable of measuring it. And so we're able to get those insights to make improvements in both healthcare and just diagnostically.
Well, thank you for sharing all of that. I have just a few more questions for you. What inspires you?
When I think about that, I think probably the biggest thing is I enjoy um, the challenge. I think I love the challenge. There's a statement I'll say that the thrill of the kill is second to the challenge of the chase. So for me, you present me with a problem, you present me with an obstacle. I love chasing a solution. I love the challenge. That excites me. That wakes me up. Where most people feel crushed, oh man, life's hard. I'm like, no, man, this is a chance to test your mettle. And so I love the challenge of it. I mean, the success is great, but I love the challenge of it. And man, honestly, in clinical diagnostics, when you're navigating both insurance, billing, federal, private, you've got constant changes and regulations. You've got all of these pieces. It is fraught with challenges. So it's an opportunity there'll be a million, if you solve a million problems today, there's going to probably be a million more tomorrow that you get to work through. And so in that aspect, it allows me to keep my brain busy and constantly looking for ways to innovate, because when you're the small guy, like we're not LabCorp Quest, we're a smaller lab. So for us, it's all about how can you innovate? How can you come up with a creative solution to a problem that others don't have? And when you've got that, that's how you do it. Like, so one of the things I did was we married up artificial intelligence into our server, and basically we loaded everything up. And so now I have a dashboard that gives me real-time feedback of raw sensitivity on the equipment. So now I'm building out predictive responses when a PM is going to be needed on a mass spec before it happens. Because oftentimes when you're using internal standard or isotopically labeled internal standards, you're measuring the ratio. And so you're still going to get a QC that passes, but that doesn't mean your signal's not dropping off. So what I did is built up an AI model that's feeding the raw signal off the instrument into the AI, and we're able to begin predictive issues of when it's going to happen. So you know preemptively, let's go and do the maintenance now. The other section that we built the AI in was on our aggregate. So all the samples coming in, we take samples from probably 10 different states and probably 200 or 300 different doctors all over the country. And so one of the hardest parts is trying to assess what's the dollar value to that account, and what if the doctor changes his ordering habits? What if he went on vacation? What resources are you committing there? And so what happens is the AI feeds in, and I'm getting real life trends on what the payer breakdown of every account as it's coming in, the aggregate Medicare dollar value of every account. And so basically I have dashboards now that I've created that I can map any doctor at any given time, and I can see, okay, well, look, you know what? I need to send the sales team out there to go talk to him. For whatever reason, the doctor's dollar value, let's say he was doing $40,000 a month and now he's doing $20,000 a month. Is it that he doesn't like our service? Is he just ordering small? Was he on vacation? And I have warnings and parameters set in that will actually tell me. And then I'm sending an email to the rep. I'm like, hey, you need to go talk to Dr. So-and-so because we're seeing this. And it also allows me to forecast, because most collections are about 60 to 90 days out. So being able to create solutions that are not just scientific in nature, but whether it's technology, the aggregation of data, and then being able to utilize it, I love that opportunity. And to me, that excites me. And so that's probably the more inspiring part of it.
With AI and automation, I personally have about 20 different apps that I've developed for our business as well, and a similar sort of thing, right? I mean, you see a problem, it's pretty easy to keep kind of plugging away at whatever the problem is, and eventually you have a solution that at the end of the day is unique to your own organization, that ultimately provides greater efficiency, but also better answer overall for the people that you're trying to serve. So I'm completely behind what you say.
Oh yeah, and that was us, as we were trying to do it manually. And when you were small and you only had one or two accounts you were looking at, it was possible. But suddenly, as you've got samples flying in different directions, you're trying to track it, and then you're wanting to reconcile to find, okay, did the insurance pay, or is it here? Where is it at in that process? And so for me, we were doing it manual for a while, and I was just like, man, this is terrible. I was like,
Yeah.
one, it was like while I would get the answers, I spent so much labor trying to get to the place where I had the information. You're almost exhausted from like, okay, now I got to make a decision. It was like I spent 3 days just getting the data together. So
Right.
for me, it was like, how can I build a system so that I can almost, and as almost a command control center, go, okay, this is what's going on here. This is what I need to fix here, and so that I could create priorities. And so that you're right, it's necessity breeds invention. And so for me, it was how can I make the job of whether my scientist, one of my people, how can I make their job easier so they can do more? How can I get better feedback? And so just that continuous process of improving things.
Yeah, absolutely. Kris, what concerns you?
I would be honest, one of the biggest concerns, and this is something that just happened last year, is overregulation by the government. And I'll give you a perfect example. So at the end of the Biden administration, the FDA said, hey, we're not going to let CLIA look at lab developed tests anymore. We're going to punish it. We're just going to be FDA. Man, anybody like how many people felt they did a good job on the COVID vaccine? Yeah, they pushed that one through, but that was also $1 billion companies that were going, here, FDA, have a little money. So what that tells me is that the FDA was going to regulate every new lab developed test. That means that there won't be any other small innovative people to be able to step into the market. And if they're going to limit me from being able to do that, then it's almost like saying, that's awesome that you were creative, that you created these solutions. But guess what? You're going to have to stop creating solutions because the $1 billion Roche, those companies that are going to build the FDA or the RUO kits for you, that's where you're going to go buy it from. And so that type of overregulation where CLIA has been looking at these tests for 50 years without a problem. LabCorp, Quest, everybody is using lab developed tests. CLIA has come in there. They've got guidelines. The CLSI, the Clinical Laboratory Standard Institute, sets out the guidelines of what you should have, your parameters that you have to make an assay meet. And then when inspectors come in, that's what they're looking for. Okay, what's your precision? What's your sensitivity? What's your interference? All the different things. So to all of a sudden go, let's overregulate it and give the FDA an authority that they've never exercised in their existence
Mm-hmm.
and say, you know what? No lab can enter the space with a new diagnostic test unless the FDA says it's okay. And you want to do a 510K approval. I think that becomes a danger. That and like the PAMA, the Protecting Act of Medicare, where they're constantly trying to reduce the reimbursements. Both of those are kind of problems. But I honestly feel the overregulation and allowing the FDA to step in and trip up innovation is going to be more problematic because let's say you've got other labs like me that are they're not the big box lab. They're going to do it by brute force, and we come up with something like I always tell people on an individual test, LabCorp can probably run it cheaper than anybody because one, they're at scale on volume, and when they design their assays, they design it to do this one test. Like I want to do testosterone as cheap as possible. I don't care about anything else. Now, at the same time, if you were to say who can do these 20 hormones like us cheaper, I'm going to crush LabCorp and Quest because of the way I designed my system. And if the FDA steps in and goes, great, that's awesome that you're creative and that you're making a better way forward and it's going to improve patient care. But unless you can go through the whole 510K regulatory thing and have us come and look at everything and spend the next 5 years waiting for it, then you can't have it anymore. I think that is going to be, and there's already some lawsuits that have been drawn into it over this. Honestly, what that really looked like to me was it was like Medicare, which kind of oversees CLIA, and the FDA fighting over our tax dollars.
Yeah,
It was like, who gets the money to go inspect the labs?
sure.
It's really our tax dollars, and they're like, we're going to take FDA wants your tax dollars. No, CLIA wants your tax dollars. But in the reality of it, I'm hoping that some of those type things back off because you'd really like to be able to keep an innovative approach. Because when you look at a lot of clinical diagnostics, I mean, there are hospital systems that are still running Windows XP.
Yeah.
I mean, so obviously, if there's a place that needs to be innovated, that needs to be technologically updated, I believe it's the clinical lab space. I believe the clinical diagnostics. I think there is a lot of room without compromising anything that you can make huge improvements that will improve patient care and offer a better insight in diagnostics. But if you're saying, well, you just can't create it, then that to
Yeah.
me seems like you're either picking your winners and losers and saying if you've got enough money to lobby the FDA, then you can get approved. But if you don't, maybe you should go work for somebody that can lobby, is what it kind
Sure.
of looks like.
An interesting challenge for sure. Last question is, what excites you?
Um, I like trying to find new tools. I'll be honest. One of the things that I've learned and like overall in leadership was a couple things. Was one, you hire your weakness and focus on your strengths. My strength is problem solving. My strength is coming up with a new system. I am not a manager of a system.
Mhm.
Like once a system's in place, I would rather go get a root canal than manage a day-to-day system. Now, if there's a problem with it, I'm Johnny on the spot. I get a text from one of my scientists. Hey, Kris, the instrument's on fire. Man, I'm there. I'm gonna rebuild it. I've taken our mass specs apart to pull even the quads out of it, rebuilt them, put it all back together to keep it up and running. But once that system is working like it should, I don't really want to look at it again because to
Mhm.
me it works. So
Right.
if it works, and I finally understood, Elon Musk made a. I was listening to a thing and he was talking about, and he said being an entrepreneur is kind of like chewing on broken glass and staring into the abyss.
Right.
And it's 100% true because like as a CEO, the only problems I'm going to chew on are those that are threatening the company. Because if somebody else solves it, whether it's my operations director, whether it's one of my scientists, then I'm not going to hear about it. So when that problem makes it to my desk, you're chewing on broken glass because it means that if something's not done, something is going to go very wrong. The other part is you're staring into the abyss because you can't really see what's next, and you're trying to fumble your way through the darkness, and you're like, is this going to work? Oh no. Just like us, I thought when I created that first finger stick, I thought doctors were going to be beating down our door to run a finger stick test. And every doctor was absolutely amazed. They were like, that's the coolest little gadget. And then the doctors were like, I don't really care if you had to take the blood out of their eye socket. I just want the results. And I was like, oh. So eventually, when we created the device on the shoulder where it was easy, doctors are still like, I don't care how I get the blood. But then it opened it up for patients and it became real easy to collect. And so the consumer liked it better. But yeah, no, that staring into the abyss, I think that, but honestly, I find that there's a certain tolerance if you're going to be an entrepreneur. And I find that when you have that type mindset, when things are cookie cutter predictable, you're bored out of your mind. And that's typically me. If everything worked like it was supposed to, I really don't know what I would do. And I think the what really excites me is health, being able to help people. I got a good perfect example is I use my science. I probably monitor my blood weekly, which is OCD to a level that most people can't fathom. But because of my diet, I went through two shoulders. I had both my shoulders reconstructive surgery to have both of them repaired, both rotator cuffs while I'm running a business. And I mapped it out. I worked with functional medicine doctors. We created all kind of therapies that allowed me to recover to the point now. What I'm getting ready for is with Core Health. I'm getting ready for the Mr. Louisiana bodybuilding contest in May, and so the goal is to be able to show all of the science, these different things that we've been able to biohack. Like we've studied when you hear about people talking about cold water immersion. Well, we've actually done the blood work. I can tell you exactly what's happening because we've actually measured it. Why can it burn fat? What can it do? And so that type of stuff excites me. I love working with doctors that are on the cutting edge. They're looking for new ways. They're looking, and I'll call them up. I'll call one of them and say, hey, what do you think about this? And they're like, oh, it could do this. And I'm like, how would we measure it? Or what do you think? And I'm like, and a lot of times they'll prescribe it and I'll look at it. We'll go through a trial and we'll like, okay, what did we see here? Did we get this? And what do we improve? And so we're finding almost whether it's natural plays, supplements, nutrition, things that you can do to really improve your health at a different level. And that's what excites me. So having the lab and the technology come together and allow me to be able to put this out where you've got a whole new way to almost the next step in human evolution, if you will. That's what excites me. And so it's almost like the lab allows me tickets to the show.
Very cool. Well, thank you, Kris Franklin, for being on the Life Science Success podcast. I greatly appreciate you being here and for sharing more or less both the journey as well as just that overall interface between medicine and the lab, which I think is absolutely so critical as well. So thanks a lot for being on. I appreciate it and wish you all the best.
I appreciate you for having me.
Thanks a lot. Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.